the nuclear transfer experiments. I summarized all of those, as I mentioned in the last. Maybe I did. I don't remember. But you know those nuclear transfer experiments were a backbreaker to this whole view that cancer is a genetic disease. You know, these studies were done by Dr. Kinnell at he was down at Louisiana. He since went to Minnesota. He passed away a couple of years ago. McKinnell's work on the frogs, and then there was Jim Morgan's group at St. Jude's, and there was Rudy Yanish's group down here at MIT, Beatrice Mintz's group, Israel and Schaefer's group from the University of Vermont, Shea and Wimberlin from University of Texas. I mean, all of these groups had done these kinds of experiments where they would take the nucleus of a tumor cell and put it into the cytoplasm. They took the nucleus of a nucleus of a tumor cell, put it into a normal cytoplasm, and you would get no dysregulated growth. On the other hand, if you took the normal nucleus and put it into the tumor cytoplasm, you got dysregulated growth. And I documented how these studies were done over and over again, repeated time and time again. Okay, all different types of cancers, all different types of protocols. And as I said, what were they doing? They were simply asking whether or not a tumor nucleus could direct normal development or not. And the answer was the tumor nucleus was suppressed in its abnormal growth. So the cytoplasm was controlling the cytoplasm was controlling the regulation of growth. And that's exactly what I said, because in the cytoplasm are the mitochondria, and the mitochondria control the cell cycle. And if the mitochondria are damaged, it doesn't make, if the nucleus is so you'd have all these nuclear gene mutations, and you put it in a normal cytoplasm, and you get normal regulated growth. I have a picture of it around here somewhere. I should have had it, but the picture says 1,000 words. You can follow the picture. I published it in this paper here. The folks can read all of the details of these experiments if you go and read the detailed documentation of these with all the references. I even have it in my book. I have a whole chapter on the nuclear mitochondrial transfer experiments. Also, if you have a cell that's dysregulated in growth, and you say, well, we're going to take out clean out the bad mitochondria and put new mitochondria in, the cell becomes regulated in its growth. And then if I have a slow growing or a normal cell and I put abnormal mitochondria in there, it becomes dysregulated in its growth. So it's just everything is controlled by the mitochondria in the cytoplasm, not by the genes in the nucleus. So this is the key that tells us that cancer cannot be a nuclear-driven genetic mutation disease, and that is a mitochondrial metabolic disease, and that changes the whole playing field about how cancer should be treated, managed, and viewed. And once that comes around, the whole system will change to a more rational way to treat this without toxicity.